Next speaker is Keith Dadson, who is working with Filio Bilia and Vivek Rao, and he's going to talk about the control of cardiac growth and energy metabolism by ubiquitin ligase. Thank you.
Hi, so my name is Keith Dadson. I would like to first thank the committee for putting this day together. It's been very great and very educational. Also thank the committee for choosing my project to fund. Thank you very much. I'm looking forward to showing some of the work that we've done. I know that the original title of the talk was a little bit more general, the control of cardiac growth and metabolism by Mule. Since then, we've done a lot more work to understand exactly how that is being regulated. And so the new title, the more detailed, is that Mule prevents the inactivation of PGC1 alpha and PINK1 through MYC in the adult murine heart. And so, as was mentioned yesterday in the stem cell discussion, and is what has become almost a central tenet in cardiology, is that after development, the cardiomyocytes, with the exception of a short period after birth, exit the cell cycle and become terminally differentiated, and that leaves the adult heart, very limited ways to compensate for stressors, and that ideally is through increased cardiomyocyte size or hypertrophy, and in response to the loss of cardiomyocytes through either apoptosis or necrosis, the laying down of extracellular matrix or fibrosis. And while both hypertrophy and fibrosis are thought to be initially protective, we know that these are signs of poor clinical outcome, and prolonged hypertrophy and excessive fibrosis lead to decreased heart function. So then it is very important for us to identify factors that maintain cardiomyocyte homeostasis. And so one way that we can do this is by understanding what the heart's innate response to stress is. And as we know, an early response to the induction of pressure overload is the activation of what's known as the fetal gene program, characterized by an increase of the natriuretic peptides ANP and BNP. These are known to be very protective. Also a switch in the myosin heavy chain to the slow twitch beta myosin heavy chain, and also an increase in alpha skeletal actin. However, in advance of activation of the fetal gene program is the expression of the early responders, c-MYC, Jun, and FOS. And it has been shown that actually the increased expression of c-FOS and c-MYC can be as early as 30 minutes after myocyte stretch, or even a few hours after the induction of pressure overload. So we know that these early response genes are quite important in the pathway leading to cardiac hypertrophy. So what do we know of c-MYC? Well, in the heart we don't know very much, but it's been very well characterized in cancer. And so in the sum of a lot of cancer work has shown that MYC is an oncogenic protein, an oncogenic factor that increases cell proliferation and angiogenesis, as well as decreasing apoptosis and differentiation. And so the question is, what does MYC do in the heart? In 2006, Rob McClellan's group showed that knocking out MYC in the heart protected the heart from developing cardiac hypertrophy in response to pressure overload, as you can see here on the right. And later on in 2010, the McClellan group showed that overexpression of MYC by itself increased heart weight body weight ratio in transgenic animals. So knowing that MYC is playing a role in the heart, we then want to understand what is regulating MYC stability. That has not been done in terms of heart failure, but in cancer, Mule, which is an E3 ubiquitin ligase, has been identified as decreasing MYC stability, as can be seen here. And so then there is a large push to decrease MYC activation of gene sets that are not only oncogenic, but in the heart would be prohypertrophic, allowing us to reach, again, balance or equilibrium. So the purpose of my work was to interrogate Mule as a putative upstream regulator of MYC in the heart. So in order to do that, we generated a cardiomyocyte-specific tamoxifen-inducible Mule knockout mouse. And so in order to do this, we take mice carrying the floxed Mule gene and inject them with tamoxifen for 4 consecutive days. And as can be shown in this Western blot here, following tamoxifen injection, we have a decrease in Mule expression up to 1 week post-TAM. And that is correlated, or that correlates with a significant decrease in Mule mRNA, as shown by qPCR. When we look at cross-sections of the heart and stain for Mule in red, again, we can see a significant decrease in Mule expression. So what happens in these animals? Well, when we induce the ablation of Mule through tamoxifen, in the red, we see a significant decrease in survival, a 40% decrease in animal survival, up to 4 and 6 months after injection. And that suggests to us that Mule is playing a very important role endogenously in maintaining cardiac homeostasis. When we take cross-sections of these hearts, as shown here on the right, the ablation of Mule induces a thickening of the ventricular wall and an overall increase in heart weight-body weight ratio suggestive of cardiac hypertrophy. Looking at cross-sectional area using wheat germ agglutinin staining in heart sections, again, we see a significant increase in cardiomyocyte diameter. And this all correlates to a significant decrease in fractional shortening. In order to understand what is going on, we ran mRNA looking at classic markers, canonical markers of hypertrophic remodeling. And indeed, in the Mule knockout animal compared to the wild type, we see an increase in the natriuretic peptides, and also an increase in skeletal actin and an increase in myosin heavy chain 6. We then ran the mRNA on a chip, and while this is a little bit small, we took some of the most interesting findings and replicated it using, again, conventional qPCR. And what we found, not to get into all the details, is we found an overall decrease in various ROS defenders, such as catalase and glutathione pink. We saw a decrease in branched-chain amino acid metabolism as well as glutamine metabolism, decrease in fatty acid oxidation through decrease in the PPR alpha and gamma, as well as the decrease in mitochondrial biogenesis as seen by a decrease in PGC1 alpha. We also have a significant decrease in OXPHOS and glycolysis. And so while this is just a snapshot of what's happening at 4 weeks after ablation of Mule, we wanted to provide further insight into what are the early responders to Mule ablation. And so we worked backwards and looked at mRNA 4 weeks, 2 weeks, and then 1 week after tamoxifen injection. And we discovered that there are indeed factors that are decreased early in the ablation of Mule and factors that are decreased later on after 4 weeks. So some of the early responders are a decrease in PGC1 alpha, ATP synthase, part of one of the mitochondrial complexes, as well as PINK. So knowing that Mule is a E3 ubiquitin ligase, which means that it targets various factors for degradation in the proteasome, we again wanted to find out if Mule was actually targeting MYC in this process of decreasing survival and increasing cardiac hypertrophy. So again, using Western blot samples after tamoxifen injection, up here we see the decrease in Mule expression in the Mule knockout animals, and we also see a reciprocal increase in MYC expression up to 8 days after tamoxifen injection. Correspondingly, we did a pull-down study, and we pulled down MYC, or we pulled down Mule and Western blotted for ubiquitinated MYC. And in the non-tamoxifen injected animals, you see a large ubiquitination of MYC. And in the tamoxifen injected animals, you see no ubiquitination of MYC, meaning that when Mule is absent, MYC is not being targeted to degradation. So then, of course, our question was, what happens when we knock out MYC in the heart? So again, using a cardiomyocyte-specific MYC knockout animal, post-tamoxifen injection, we see a significant decrease in MYC expression, correlating with a significant decrease in MYC mRNA. However, when we looked at overall heart morphology, heart weight, body weight, and also heart function, we found that the MYC knockout animals were normal. So then our next idea was to generate a MYC and Mule double knockout mouse by breeding the two mice together. Here you can see a significant decrease in Mule and MYC post-tamoxifen injection that correlates nicely with a significant decrease in mRNA. So now the difference between our three different models are quite simply the Mule knockout has an increased expression of MYC, the MYC knockout has a normal phenotype, and the double knockout has a decreased expression of MYC and Mule. So now when we look at these animals and compare them to the double knockout animals and compare them to the Mule knockout animals, in the absence of both Mule and MYC, we see again a restoration of normal heart morphology, while in the Mule knockout that express a high amount of MYC, we see hypertrophic growth. Heart weight body weight ratio is also normalized in the double knockout animal. And also cardiomyocyte cross-sectional area. Fractional shortening is maintained. And no activation of the fetal gene program in the double knockout animals. Looking at the extracellular matrix using a stain for the ECM, as you can see here in green. In the Mule knockout animals, we see a significant increase in fibrosis. Nuclei are blue, and the clustering of nuclei in the green areas suggest the rapid recruitment of fibroblasts to the fibrotic areas. In the Mule and MYC double knockout animals, we see a slight but not significant increase in extracellular matrix accumulation, suggesting that they are not fibrotic. And corresponding with that, we see an increase in apoptosis in the Mule knockout animals, and that is rescued by the double knockout of Mule and Mick. So next, as I had showed you previously, we looked at mRNA of PGC1 alpha and PPARs, alpha and gamma. And so we wanted to confirm that by Western blot. And indeed, in the Mule knockout animals following tamoxifen injection, we see a decrease in PGC1 alpha. And again, that's rescued by the ablation of both Mule and Mick. Looking at mitochondrial complex 1 and 4 activity using a kit, we see that mitochondrial activity is increased in the double knockout animals. So just as a summary, what we've identified in the heart in an early responder to pressure overload-induced cardiac failure is a regulator, namely Mule, which will ubiquitinate Mick and target it to the proteasome for degradation. That means that Mule is a homeostatic or exerts homeostatic function on cardiac function and morphology through degradation of Mick. However, in the absence of Mule, we see a significant increase of Mick expression, and that leads to hypertrophy, fibrosis, and apoptosis. This, while this was characterized previously, what we've added is a more in-depth understanding of the process that this is taking place. So increased Mick is having a decrease on ATP synthase expression, a decrease in PINK, and a decrease in PGC1 alpha. We also see a decrease in the ROS defenders catalase and glutathione. And the decreased expression in PGC1 alpha and, of course, the ATP synthase and the mitochondrial complexes lead to an overall decrease in ATP, while a decrease in ROS defenders will have a function of increasing ROS, which begins this vicious cycle of damaging mitochondria, leading both to apoptosis but also to increase in ROS, which then causes fibrosis. So our ongoing work at this point is that we want to understand the regulation of Mule in heart failure. We know that Mick is activated in cardiomyopathies. We want to see if the upregulation of Mick is due to a change in Mule activity, especially early on, if it is within 30 minutes or an hour of inducing pressure overload. And as such, we want to identify regulators of Mick expression and activity, some of which has been done in cancer. And the suggestion is that another E3 ubiquitin ligase might be actually targeting Mule for degradation, which then leads to Mick activity. So where do we go from here? This is a very quick slide. I'll just mention briefly that ACE inhibitors and ARBs have been shown to downregulate Mick expression, but most of this work has been done either in vivo or in vitro. And so a firm understanding of how this happens, or if this happens in human patients is needed. And also targeted inhibition of Mick in coronary artery disease, since Mick is upregulated in proliferating cells, has been done or is ongoing in some clinical trials, but very little work has been done to target Mick in cardiomyopathy. So I'd just like to thank the members of my lab and also my supervisors, Viv Rao and Phyllis Bilia, and again, thank the Ted Rogers Committee.